Hi, my name's Rod Cook and I'm a medical and biomedical scientist currently working on neonate immune responses, predominantly preterm neonates and their biochemical immune reactivity. The following information is not to be taken as medical advice. It's just general information on coronaviruses and the novel coronavirus that's currently spreading from China throughout the rest of the world. For information regarding your health and the health of you and your family and your loved ones, always talk to your healthcare professional, your GP, your doctor, or pediatrician first. That should be your first line of health support always. Your doctor, your GP, your pediatrician, and so on. So first, a bit of the history about coronavirus. If you want to follow this, I'm going to stick links to all this below the video on YouTube, so you can go and follow up and look at the references and so on. So coronavirus was first isolated in the 60s by Tyrrell and Bino. This is in the UK, as shown here in the Lancet Journal of 1966, a virus they named B814. So B814 was found in human embryonic tracheal organ cultures obtained from the respiratory tract of an adult with a common cold. They successfully, although were unsuccessful at first at replicating B814 virus, however, Humray and Prochnow were successful around the same time. So Humray and Prochnow were able to grow the virus in tissue culture from samples obtained from medical students with colds. Humray and Prochnow called this virus 229E. So these viruses were termed OC, designated because they were grown in organ cultures. So OC for organ cultures. Then Almeida and Tyrrell performed electron microscopy on fluids from organ cultures affected with B814. And what did they find? They found particles that resembled the infectious bronchitis virus of chickens. In the late 60s, Tyrrell was leading a group of virologists working with human strains and a number of animal strains. These included infectious bronchitis virus, mouse hepatitis virus, and transmissional gastroenteritis virus of swine, all of which had been demonstrated to be morphologically the same as seen through electron microscopy. They all look the same. So in the image you're looking at the moment, you're looking at, if you look closely at the top, more on the right hand side, you'll see what looks like finger-like projections around a block. This new group of viruses was named coronavirus because of the crown-like appearance on the surface projections. It was later officially accepted as a new genus of viruses, as shown here, published by Tyrrell et al. In 1975 in Intervirology. In the 3 decades, yes, the 3 decades after the coronavirus discovery, the human strains of OC43 and 229E were studied exclusively. OC43 easily adapted to grow in suckling mouse brain and subsequently to tissue culture, was found to be closely related to the mouse hepatitis virus. Strain 229E was grown in tissue culture directly from some clinical samples. The two viruses demonstrated periodicity. Periodicity means that larger epidemics occur at 2 to 3 year intervals in those two that they were studying. The predominant illness associated with these infections was an upper respiratory tract infection with cases of pneumonia in infants and young adults. So then we enter SARS and MERS. So SARS and MERS are both coronaviruses. So severe acute respiratory syndrome, SARS, emerged in 2002 as a coronavirus from southern China, which spread throughout the world with a quantifiable speed. During the 2002-2003 outbreak, SARS reported in 29 countries, including North America, South America, Europe, and Asia. Overall, about 8,100 people were infected, with almost 800 SARS-related fatalities. MERS, which stands for Middle Eastern Respiratory Syndrome, was discovered in 2012. Over 2,000 cases and over 700 deaths occurred from MERS. In this image you're looking at, you can see coronavirus strains, bottom right-hand sort of corner, you can see the coronavirus 229E growing in WI38 cells. So what is coronavirus? So a coronavirus is a family of viruses. So there's not just one or a few, there are quite a few subtypes of coronavirus infecting humans, bats and peguma. Other than that, there's zoonotic coronaviruses infecting other animals. So there's feline enteric coronavirus, FECV, avian infectious bronchitis virus, turkey coronavirus, pheasant coronavirus, the beluga whale coronavirus, and even the Asian leopard cat coronavirus. So interestingly, the SARS coronavirus was linked to a bat reservoir with a possible intermediate host being the palm civet, which is an omnivore native to South and Southeast Asia. And the MERS coronavirus was linked to a possible bat reservoir with possible intermediate host being the dromedary camel or the Somali camel. So while on SARS and MERS, the mortality rate for SARS and MERS stands at around 10 and 35% respectively. So don't forget there's links below. These coronaviruses are categorized into alpha coronaviruses, beta coronaviruses, delta coronaviruses, and gamma coronaviruses. And just for the sake of the length of this video, you can look up the difference between these. I'll put some links below. So using this recent study in the Lancet, a 2020 study, let's look at the infectious coronavirus currently in the news in China and around the world. So the article, a familial cluster of pneumonia associated with the 2019 novel coronavirus indicating person-to-person transmission, a study of a family cluster. So there's currently genetic data and information on 33 distinct corona subtypes. So 13 of these subtypes mainly affect bats, 18 subtypes affect mainly humans, and one subtype affects mainly peguma. You're looking at a list now of the different bat subtypes, the different human subtypes, and the peguma subtypes. Interestingly, I put in red the current coronavirus serotypes or subtypes in red. It's not just one, there's quite a few. So it's like dogs, there's different dogs, they're all different. They might be similar, but they're different. Same as coronavirus, they're pretty much similar, but they're different. So there's slight differences between all these different coronaviruses. So looking at it, there's quite a few different ones, and they're all sort of genetically related to each other with some slight differences. So what do we know about these coronaviruses? So there's single-strand RNA envelope viruses with pleomorphic morphology. So pleomorphic just describes its shape or size and the blob-shaped shapes. So you're looking at a picture on the right, the left-hand side under coronavirus is a picture of coronavirus, or what it looks like sort of under the microscope. You've already seen that anyway. Okay, so the envelope viruses resemble the morphology on the right hand side of that image in this that you're looking at now with the little purple blobs around it. That's sort of what a coronavirus looks like grammatically or diagrammatically. Coronaviruses are typically 60 to 220 nanometers in diameter. They are positive stranded RNA with a helical symmetry. So positive stranded RNA can be directly read by the ribosome and therefore can replicate without the additional steps required by DNA and negative sense RNA viruses. These include a short video link below so you can look up that. The coronaviruses have characteristic club-shaped projections of around 20 nanometers involved in neutralization, fusion, and in some coronaviruses even hemagglutination. So some human coronavirus strains such as OC43 and 229E have been demonstrated to have an affinity for neural cells, so brain cells, and persisting in them, possibly causing chronic disease. So antibodies to OC43 have been detected in the central nervous system of patients with multiple sclerosis and Parkinson's disease. So OC43 RNA has also been detected in human brains and has been demonstrated to infect astrocytes, and microglia. Acute and a chronic infection of OC43 was tested and demonstrated in neural cells such as oligodendrocytes, astrocytes, microglia, and neurons. So there's an article I've linked below that talks about neural persistence, meningitis, encephalitis, and chronic disease associated with coronaviruses. So various human coronaviruses are responsible for 10 to 30% of all common colds. They can be detected by indirect immunofluorescence, ELISA, or reverse transcriptase PCR confirmed by Sanger sequencing. So reverse transcriptase PCR is what's being currently used on the outbreak in China and around the world. So all age groups are affected by coronavirus. Infection rates have been shown to be uniform for all age groups. So this is not an infectious disease for one age groups like the elderly. It's also infecting teens, children, adults, and elderly alike. So with the pathology of coronaviruses, they affect the upper respiratory tract epithelium. There's a poor immune response to them. There's no cross immunity, and patients show bilateral ground glass opacity. The route of transmission is aerosol, so it's via sneeze, can be via hands, or fomites. So fomites are like via clothes, utensils, furniture, and so on. The incubation period generally for coronaviruses is about 2 to 5 days, and the duration of the disease is about a week after that. But it depends, it varies from person to person depending on their health and well-being, their diet, and so on. The severity of the disease causes little or no fever. So we're talking about all coronaviruses now, not just one. So little or no fever, nasal discharge, malaise, cough, sore throat, pneumonia, and death. Okay, so death's not minor. Death's not a rash or just a cold. So I've linked some articles that have all those details below. So I had a look at the numbers, and the numbers coming out of China, not sure whether they're 100% reliable, have around over 100 deaths and 4,500 cases. So that puts the mortality rate at about 1 in 42. So of the few thousand that have been infected, a 1 in 42 death rate is not a rash, and it's not a cold. That's not the statistics we get from rashes. A 1 in 42 death rate is around 2.3%. So to put that 2.3% in perspective, if the virus hit Tokyo and 1% of the population got infected, so 1% equates to about 375,000 people, then approximately 8,600 people would die. In New York, if 1% got infected, that's around 86,000 people with around 2,000 deaths. If it was Melbourne, if 1% got infected, that's around 50,000 people with around 1,100 deaths. And if it was London, if 1% got infected, that's around 93,000 people and around 2,100 deaths. The following table I've just put up shows the incubation periods, the median incubation periods, and the confidence index for those median incubation periods for various viruses, including coronavirus, the current one. So the normal human coronavirus on average, 3.2 days. The SARS coronavirus is 4 days on average medium incubation period, and the 2009 novel coronavirus is 3.6 days. So they vary. Coronaviruses vary from 3 days to 6 days according to this data, and I'll put links below with the incubation periods and some journal articles relating to those incubation periods. So just have a quick look at go and have a quick look at the familial cluster of pneumonia associated with the 2019 novel coronavirus, indicating person-to-person transmission, a study of a family cluster. It's got some really great data in there, some really great biochemical data, some really great statistics and epidemiological information, people that did or didn't visit markets, and so on, to give you a better idea. So just remember, the information I've just given you, most of that is fairly generic, some related to the 2009 coronavirus strain or strain subtypes. Remember, there's more than one, and the rest of it is just generic. So of the human 2019 subtypes, I've just put a list up there. So there's the genetic variant differences in the subtype. So once again, going back to my dog analogy, this would be like they're all dogs, but there's different types of dogs. They've got differences in them that make them all unique, and those differences may be minor or quite a lot, but there's not just one coronavirus. They all share similar genetics. They also share a lot of conserved domain, which is why they're all 2019 novel coronaviruses. But apart from those conserved domains, there's differences, and those differences can have a huge effect. And this just shows that this pathogen isn't just one thing, and it's quite diverse. I hope you've enjoyed this information. If you want any more information, go through the links below. Otherwise, talk to your healthcare professional or your doctor if you have concerns over anything relating to not only the coronavirus outbreak and your area, but any other health concerns. Talk to your medical professional first. Thank you. Bye.